Better watch the trout.
The experiment in Maui was to study this species, Leptocerus hawaiiensis, that grows at 300 feet, and to try and understand how these corals can survive at such depths. I had this expectation that we would see sporadic coral heads because they were living in this extreme environment at the edge of what we thought was possible, when in fact it was just a lawn of Leptocerus as far as you could see. Coral are animals that are symbiotic with single-celled algae. For whatever reason, the field has spent a lot of time studying the algae. The big questions that I'm trying to address deal with understanding the animal host itself. When we study the skeleton, they deposit in layers, just like tree rings. So there's a natural archive, a preserved record of the environmental conditions. We can understand how changes over time might affect the way coral deposit skeleton or grow. This project is being done in collaboration with folks at the University of Hawaii. To work at those depths is very tricky. 300 feet is about the maximum limit that scuba divers can work. These are technical divers that work on rebreathers. And it is at the shallower limit of submersibles. The submersible has a big robotic arm, not very dexterous. That arm breaks these very easily. It can't delicately choose a fragment or not, whereas the scuba divers can. The scuba divers only have 20 minutes of bottom time at that depth and then spend 2.5 hours decompressing so that they can do one dive the next day. Divers can collect the coral fragments we need, place them in the experimental chambers, start the experiment, and then head to the surface. The submersible takes over, and that's the part of the experiment I was involved in. I was in the submersible. Can then spend the extra 3 hours needed at the bottom and then remove the coral from the chambers, take samples, bring those to the surface. So by taking advantage of the long bottom time of the submersible with the fine motor skill of the scuba diver, we were able to execute an experiment that would otherwise be totally impossible. We met every goal we wanted to, and now it's a matter of analyzing samples we brought back to answer the questions we have about how are these corals managing to live in those environments. Coral are extremely sensitive to very small changes in temperature. We're starting to worry also about the acidification effect of increases in atmospheric CO2.
So the oceans are getting more acidic, and we want to simulate that in the lab according to values that are predicted for the year 2050 and the end of the century. And so we need very sensitive pH probes, and so that's what we are testing here in the lab to get the system working.
We have another funded project that we're doing to look at that specifically. It just started. And we're doing it in collaboration with Todd Mehlman at the Coral Reef Farm in New Albany, Ohio.
This is a huge opportunity for me to work with Andrea Gritali at the Ohio State University. They know a lot more about the chemistry and about what's actually happening, obviously in the oceans and with the animals. They get to see it on a regular basis. So being able to collaborate with them and to learn more about the sciences, it lends for an opportunity for me to build my business even further.
The School of Earth Sciences represents a very interdisciplinary background for me to do my research. There are linkages between the university and private sector. The opportunity to work with the Coral Farm is encouraged by Ohio State University. Coral reefs are often viewed as canaries in the coal mine. They seem to be the ecosystem that is taking the hit the hardest, the earliest, as climate change continues to charge forward. Our research suggests that there's some hope that there are species out there that in fact can adjust to repeated stresses. The overall state of reefs will go down. How badly that is going to be and whether it's complete devastation is where the questions lie.